everyone. Welcome to today's live broadcast, Controlled Release Redefined, the strategic use of excipients in formulations. I'm Shannon Stoltz, Senior Editor of Special Projects for Pharmaceutical Technology, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by IFF Pharma Solutions. IFF Pharma Solutions pioneered excipients like Avicel and Methylcellulose nearly 70 years ago. It has since created a unique range of well-known excipients designed to deliver performance and cost advantages across oral dosage, oral liquids, and topicals. The company's commitment to quality, service, and reliability isn't just to check a box, but to drive an endless pursuit of delivering the right answers at the right time. Before we get started today, we do have a few quick announcements. First off, this webcast is designed to be interactive, so we encourage you to ask questions throughout the event. Any questions you have can be submitted by typing them into the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player, and keep in mind that the slides will be advancing automatically during today's event. If you have any technical problems, either viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. Moving on, I would now like to introduce our speakers for today. We are very pleased to be joined by True Rogers and Adriana Quiroga. Dr. True L. Rogers is a Senior Lead Scientist and Technical Fellow at IFF Pharma Solutions, and since returning to R&D from his technical service responsibilities in Germany, Rogers has spent the last 14 years driving innovation and growth. This work includes launching innovation platforms as well as supporting customers in their customization, development, and problem-solving journeys. He's recently added biologics collaborations to his repertoire with the development of several projects in this landscape. Rogers is a thoughtful mentor to many scientists across the globe, providing project and personal guidance. He is a licensed pharmacist, earning both his Bachelor of Science degree in pharmacy and Doctor of Philosophy degree in pharmaceutics from the University of Texas at Austin. And Adriana Quiroga is a successful pharmacist with over 20 years of experience in the pharmaceutical industry. Her expertise lies in providing crucial support for the development of oral dosage forms from initial concepts to production. Quiroga has dedicated nearly 2 decades to working in the field of modified release formulations with a deeper approach to matrices. She is an accomplished researcher and has authored and co-authored several posters that were presented at several Latin American congresses, CRS chapters, and conferences for the Association of Pharmaceutical Scientists. Prior to her current role, she worked at a distributor offering technical support to customers. And additionally, she has also served as a professor in a postgraduate course at the University of Belgrano and the University of Business and Social Sciences in Buenos Aires, Argentina. Her contributions to the pharmaceutical field continue to make significant impacts. Thank you both so much for joining us today, and please get us started.
Thank you.
Okay, thank you, Shannon. Let's go to the presentation. True will make the favor. Okay. Well, good morning, good afternoon, good evening, everyone, depending on the part of the world you are. Then in this presentation, we'll talk about control release because who don't struggle with control release formulas in some way. And we will go with exploring in a deeper way the excipients that we can use in an extended release formula. Going through the agenda, please the next slide, True. We will see like the agenda is based on 2 main areas. One will be more introductory. We will talk about IFF portfolio and capabilities in a brief mode, should you understand how we can support you. We also talk about the control release, why we are going through control release as a formulators, and the different or most common ways of achieving a formulation of control release in the area. And hydrophilic matrices will be like how it is an hydrophilic matrix, how it acts, the mechanism. That would be like introducing part. And then we go with a deep dive on the considerations of use of HPMC in matrices, actually methylcellulose with True. And then, of course, we are saving some time, as Shannon mentioned, for a question, then I encourage you to go to question and answer box in order to put this question and make it more interactive. Then let's start talking about pharma solution at IFF. In the next slide, please. We will see that we have a big footprint in the industry with 1,600 employees globally, 12 manufacturing plants, and we have 8 technical centers of excellence for focus on research and development, where we explore more in a deep way the chemically and the new potential products. And in the other 4 that are application development and innovation centers are more focused on supporting our customers in applications in a special project you are trying to get. Then if we go to the next slide, we can see the global footprint. Where are we located? And you can see, I will not go for all the spots, of course, because it's a lot, but you can see in white the manufacturing plants. We have in US a couple of centers there of production, different products, Europe, and some in Asia Pacific. And in green, you will see the technical excellence centers. As I mentioned, we have like two types, and you will see that in some cities we have the both. That's the reason, because there are not 8 there. Then we are very good equipped for supporting your project. Then moving to the next slide, let's talk a little about the portfolio. Of course, that pharma solution portfolio is broader than this, but we will focus in this topic of today on control release. Then talking about control release, the most used pharmaceutical form are matrices, and then we are plenty of product for supporting that development, as Metocell, HPMC, could be methylcellulose, but basically HPMC, polyethylene oxide, ethyl is ethyl cellulose, and we have different kind of alliginate and pectins to make it, to give you a lot of options to achieve your profile. For barrier membranes, we have the 2 options. One would be aqueous, and the other one would be organic, ethyl for organic application, and aquacoat, it's a latex for aqueous application. And for osmotic pump, we have like the main player in osmotic pump, that is the polyethylene oxide, and could be also use Metocell. Then let's go deeper on the fundamental on control release. If you go to the next slide, thank you. Here we can see, in this case, we are in the graphic, we are seeing the plasma concentration, like in vivo situation, right? Plasma concentration against hours. Then we have 2 dotted lines that make a therapeutic window, and that means that below that we don't have any action of the active. For example, I don't know, antibiotic. We don't have any antibiotic in action. And if we go to the upper line, above that in the threshold, we will start having side effect or even toxic, depending of the drug and the concentration. Then if we go to the different lines there, depending on how it's set in your screen, but like we have the first one, that is the oral disintegration, that the light green one, that is actually a modified release also, because you have absorption in the mouth, then it's quicker than the immediate release, that is the stronger blue. We have an immediate release, all the releases in once, then it's absorbed, and then by clearance is going down. Then we have a time in the therapeutic window, and then we have to take another dose in order to complete the treatment and to be in the therapeutic window more time. Then the other line that is like a light gray, that is exactly the same shape that immediate release one, but this like delayed in time, actually is delayed release, and in this case could be enteric or colonic. We are like targeting the place by pH, for example, or timing of opening the tablet, but we are not controlling the rate of the drug going out. Then in this case, we'll be in a different place, but actually all the drug will go out, we have the clearance and go down with the time. Then we have the ones that we were focused on this presentation is the extended release. That is the one, like, yeah, the color is like a gray, and it's in the therapeutic window for more time, and that is really the objective of this formula, having just one dose and staying in this therapeutic window for more time, making the life of the patient easy. Then if we go to the next slide, we will see the other way around, the way that we probably are more used to see it, that is the in vitro profiles. Then talking about modified release, extended release formulas, the two kinetic more usual are first order, then when we have release of the drug quickly at the beginning, and then we have the control. That is the most used, the most common profile in HPMC matrices. And I will talk a little about this, and within the true part, you will really see why. And zero order, in this case, we have a rate, equal rate all time, right, of a drug going out. And in this case, it's most related with osmotic pumps or probably some barrier membrane pharmaceutical forms, like a multiparticulate, for example, are easy to achieve. Are not zero order is not impossible to achieve with a matrix, but it's challenging depending on the drug solubility. And then we have another profile, well, enterics we already talked about. And pulsatile, it's really challenging, it's innovative, and it's really difficult to achieve with a matrix itself, but probably combining different layers or different multiparticulate population in the same pharmaceutical dose, it's possible to achieve. Then what are the advantages of using control release? In the next slide, we will see that the main advantage is related with the patient compliance, because if you take into account that in the last years, it was not so many new actives, new API, new chemical entities, I mean, the developments are more focused on how to release it, how to reduce more side effects. And then for patient-centric development, modified release is a great tool. Then, of course, reduce the dose, reduce the responsibility of the patient, because, you know, you have to take just one a day, for example, especially for chronic disease or for medicated persons, like, you know, our population is aging and we have a lot of patients with a lot of medication, then it will make the life easy. And then, of course, because it's more control, we are less exposed to be in the toxic area, in the side effect area. For marketing reasons, also it's a good tool. You can extend the life cycle of the drug, and it's pretty good for this reason. For what are the disadvantages? Actually, the cost, depending of the pharmaceutical form and the mechanism we are using, could be really costly or not so, depending. Also, because we have more hours of correlation in vivo, in vitro, that could be a challenge depending how the release of the drug is done. That could be a thing to take into account, the correlation in vivo, in vitro. And also for patients that are used to crush the tablet, chew the tablet, that is not possible with these pharmaceutical forms. Then going through the next slide, we will see the different, actually, there are a lot of different type of approach for having a modified release, extended release, but focus on the most used. Of course, matrix is far away the most used pharmaceutical form for extended release application. Hydrophilic is more used than inert. Then we will, in the next slide, talk a little about this, but in general, no, sorry, let's move. In general, it's okay. Hydrophilic matrices are, the complexity is low because you use the same tools that you have for making a tablet. Then, you know, there is a lot of knowledge about it. But in the release, we have diffusion and erosion. Then the in vitro correlation is challenging. We will go deeper in some slides that are ahead. Inert matrices the concept is the same, but in this case, the polymer don't hydrate, and we have a more complexity because is more dependent on, for example, the process. If you put more pressure for make the tablet, probably you have variability that in an hydrophilic matrix is not sensitive to that. For barrier membrane or multiparticulates, we have diffusion as a release. Then you have different ways to achieve a multiparticulate. The most used is drug layering, and that's a reason because you can really load a lot of drug, but because the production times, it would take ages. Then in general, the dose could be low to mid because the production times, it's more complex, it's really have more cost because you have some equipment that probably you don't use for a matrix or a common tablet, but have better in vitro correlation because it is through diffusion how the drug goes out. And finally, osmotic pumps are like really great in terms of correlation because you only need osmotic pressure that is constant in all human beings, but it's more complex because you have to have the knowledge and the equipment to do that. Then that is why it's not so common use. Then now, yes, please, let's go to the next slide that start talking about the matrix concept itself. This is a graphic that shows like a tablet have a lot of different excipients and drug, but in this case, let's focus on the two main players that will be the drug and the release modified polymer. In this case, I used to think that it's a kind of spaghetti, right? And we have this mix in the whole mass. Then we have in the surface. Surface is a word very important for matrices because it's a surface action that we will have as drug will go out. Then if we go to the next slide, and it is an animation, I hope it works. Then we have a tablet with this drug and polymer. Then in contact with water or with fluid, we will see and start wetting all the surface. Then in this case, if we have HPMC as a polymer, then we will sew a swelling layer. And all the drug that was in the surface, if it is soluble, goes out. Then like a burst effect initial, all the drug going out quickly. Then with the time, this gel or this swollen layer actually will be stronger and will be more hydrated, and they start controlling the drug to go out. Then if the drug is soluble, we'll have to go and diffuse for this layer. If the drug is insoluble, have to wait until we have erosion and the tablet finalized goes, erode completely. Then it's very important, the swollen layer, how it's made, and True will go really on this detail, how it's made, how strong it is. That is key for a extended release formulation in a matrix concept. Then in the next slide is a simple graphic, but for me it's easy to explain to you how is the release. Actually, it's the same, but going with a loop. And we can see like two scenarios. Soluble API in blue and insoluble API in red, something like red. Then we have the spaghetti, we have the drug there inside. With dry polymer, everything stays. When we start the hydration, the water comes in, and in case of dissolvable, will dissolve the drug, and the drug will diffuse for this for this layer and goes out to the media. In case an insoluble API stay there, cannot be dissolved, and then have to wait until the polymer unroll at the end, totally hydrated, and go to the media. Then with more water, probably we have more chances to the drug to dissolve at the end of the dissolution time. Then this is a mechanism. Insoluble will be by diffusion mainly. Soluble diffusion mainly, insoluble by erosion. But of course, will be a combination of both in terms of the intermediate point of solubility. Then moving to the next slide, we will see a typical matrix and what are the main players on a matrix. It's not working. Yeah, working. Then we will see API, of course. I say, well, we have like two main players. API is the diva, right, of the decision tree, is the fundamental stone to take the other decisions. Then if it is water soluble or if it's poorly soluble, we will decide which type of polymer we will use. And of course, with the polymer, there is another option that I have. True, we'll go deeper on that. I will not take this in here, but it will be hydrophilic, hydrophobic, inert, will be different concentrations can be used. In general, we are targeting 20 to 35% depending on the polymer. Then we have two important small excipients that we have to use, lubricant and glidant. We need to ensure that we will have a good mix and good flow to avoiding have different a lot of deviation in weight, for example, that doesn't help on the matrix tablet. And then the dry binder actually can help us to make a fine tuning. It's not really a big impact, but if you use water soluble or water insoluble, can give you a different strength of this swollen layer, then can give you this opportunity to fine tuning on the formula. Then summarizing why HPMC, hydrophilic matrix, it's important in the next slide. As I said, actually it's for making a modifier release product in an hydrophilic matrix, you have to use exactly the same tooling that you have, like tableting machine, mixer, tableting machine. The worst case, you have to granulate. The granulation method could be high shear, fluid bed, wherever. Then in that case, it's really important that you are not have to invest in another equipments, are inert, have a lot of history in the pharmaceutical global market. We really know that are safe, are stable, and actually it's important because we can achieve a robust formula. But of course, if you feel, if you follow some important step that we will explore in the next part. Then say that I will pass to Drew came along to the Mariana Trench of the knowledge on HPMC methylcellulose use. Drew.
Thanks so much, Adriana. You gave me actually a great foundation to spring off of. So Adriana set the stage, very well. What I will be focusing on are the structure, property, performance relationships, right? So what is it about the cellulose ether that enables controlled release functionality? So hopefully, as I take you through this second half of the presentation, you'll start to see how the pieces fit together. As the title shows, these cellulose ethers that we will talk about are water-soluble cellulose ethers. They are derivatized cellulose materials. You can see one of these 10 hydroglucose units of this cellulose backbone is what native cellulose looks like. At the 2, 3, and 6 positions, you see these hydroxyl groups. Well, what we've done as polymer scientists is we have derivatized some of these hydroxyl groups to where you have a methyl ether on some of them, or you have a hydroxypropyl ether on some of them, or you have a combination in various ratios and concentrations, extensive of substitution. both methyl ethers and hydroxypropyl ethers. These give you the different, cellulose derivatives that you see in this grid on the lower left-hand side of the slide. For example, if it's just methylcellulose, it would be in that lower right-hand side of the figure, that orange bar. We call it sometimes 2900, for methylcellulose. That's because if you look on the X and Y axes, that's methyl substitution and hydroxypropyl substitution, respectively. 2900 means it has nominally 29% of those hydroxyl groups are substituted with a methyl ether, but no hydroxypropyl ethers. We'll talk a lot about 2208 or K chemistry. It's this black box kind of in the lower left-hand side of that grid. If you look towards the center of that, you look at 2208, 22% nominally is methyl substitution and 8% nominally is hydroxypropyl substitution. So again, it just kind of gives you a tie of the nomenclatures that are often used with these water-soluble cellulose derivatives. But really the name of the game for hydrophilic matrix tablets is, formation of that hydrogel, as Adriana was talking about, that envelops that matrix tablet. What critical attributes of the polymer enable formation of that hydrogel around the matrix tablet? And we will talk about those and step through them in progressive fashion. So we talked about the different chemistries. Here's an idea here. Here's another grid of 2910 or E chemistries, different molecular weight grades along the x-axis. Along the y-axis are the different chemistries themselves. It really depends on the application in which you're using, or the formulation and the delivery system in which you're attempting as a formulator, as to what chemistry you may start to try to use. If it's a film-based application or a solution-based application, it might be one of these F chemistries or E chemistries, in particular the lower viscosity grades. If it's like what we're talking about within the scope of today, a hydrophilic matrix, well then really a K chemistry of these different molecular weight grades is going to be the workhorse. That doesn't mean you can't formulate with an E chemistry. I have worked with formulators in developing E chemistry-based matrices. I can even think in one time in my career, I've worked with a formulator on an F chemistry hydrophilic matrix. But I'd say 95, 99% of the time, we're going to be talking about a K chemistry-based matrix. And I'll talk about why as we move along in the presentation. So I'm trying to kind of describe what I have in my mind in this slide, right? So I always have this pyramid in my mind when I'm helping a formulator develop a hydrophilic matrix, or when I'm developing one myself. And as I think of a pyramid, I want the base or the foundation of that pyramid to be nice and structurally sound. And then I build layers on that pyramid to make it structurally sound so it doesn't crumble, right? And I think about that as an analogy of matrix performance robustness as it pertains to these polymer functionality enabling attributes of the cellulose derivative. So at the base of the pyramid, I like to think in terms of which chemistry is going to give me at body temperature, 37 degrees Celsius, most ready hydrogel formation of that matrix hydrogel that envelops the matrix tablet. Once I have that set, then I move up to the next layer. I want to achieve a percolating network of HPMC particles throughout that matrix tablet. No matter how you slice it or dice it, right? It's a monolith, right? So I want a uniform distribution of HPMC particles throughout that matrix tablet. Once I have that, and I have robustness set from that standpoint, now I start to think about some of those aspects that Adriana was talking about. What's the solubility of the API? That may help me to move towards a lower viscosity grade, for example, with a more poorly soluble API, or a higher viscosity grade with a more soluble API that needs more diffusion dominated type release. Also, you need to think about the duration of controlled release that you're going for. 8 hours, 12 hours, 24 hours, whatever it might be. And then, I can keep going from there. At the top of my pyramid, I have, all right, so how can I fine tune release. And I might do that within the chemistry of the cellulose derivative that I'm working with. So I'll take you through this. I'll start at the base of the pyramid, moving through these next slides here. So at the base of the pyramid, I need to set the stage first by saying these are unique cellulose derivatives, right? They have unique temperature-dependent properties. They're soluble in water at low temperature. They're insoluble in water at high temperature. Normally, as a formulator, you would think, oh, I'm going to heat something up to get it to go into solution, right, to improve the solubility. Well, we do heat these up, but it's for a different reason, right, when we're preparing solutions or something along those lines. You can see here at higher temperatures, though, we either get these nice structurally sound gels that are formed in the case of like methylcellulose. You can even see in the middle picture here on the top, you can take a stainless steel ball and set it on the surface of that gel and it won't sink within, right? Or you can get these weak gels. You've heard of cloud points, a cloudy, a weak gel, which is what you'll get with a hydroxypropyl methylcellulose. That doesn't mean that these are not good, for hydrophilic matrix tablets. You have to kind of look at the whole picture, which is hopefully what I will paint for you as we step through the rest of the presentation. Well, let's keep going. At the base of that pyramid, we have this technique that we talk about called the hot-cold technique, and that's usually to prepare solutions of these polymeric excipients. We have to prepare solutions to measure molecular weight or whatever it might be, or viscosity. So we start on the right-hand side of the arrow where, in hot water, these are going to be insoluble, but that's how we disperse the powder, right? Because it's not going to be soluble. If we dispersed it in cold water, well, then it would clump together and create what we call fish eyes. And well, that takes a lot longer to get a solution. So if we disperse in hot water and agitate, then we cool down, then we get our solution, right? And once we have our solution, let's say we heat back up. Now we get that gel or precipitate that you saw in the previous slide. What if we cooled back down again? You go back to solution. What if we heat back up again? Then you go back to your gel or your weak gel. This is what, and so on and so forth. This is what we call reverse thermogelation, right? These are very unique polymeric excipients in that they have these unique temperature-dependent properties. Well, let's think about that from a rheological standpoint. And I paint it on two sides of the coin. On the left-hand side, I look at solution rheology. So you start with the polymer dissolved in solution, you heat up in controlled fashion, and then you go through various thermal events from solution rheology. Here you're in a rheometer, you're looking at storage modulus or G prime. If it's a methylcellulose, you get these strong gel formations at certain temperatures as you ramp up the temperature. If it's a hydroxypropyl methylcellulose, you have a precipitation event that occurs. So you see a drop in the G prime, and then you see a recovery of that G prime as the weak gel forms. Okay. And, depending upon your chemistry, you have a certain fingerprint of a solution-based rheological profile that you can see here that's unique to those chemistries. You might think to yourself, well, then why is K chemistry so prevalent in hydrophilic matrix tablets? It has a weak gel, it gels at a high temperature. You have to think about the other side of the coin, which is the right-hand side of the slide. In this graphic, this is what we call powder dissolution temperature in our labs. This is a technique that my friend and colleague in Bomlitz, Matthias Kanar, he and his team have developed and perfected this technique over the years. There are actually many types of rheological techniques, but this one we call powder dissolution temperature. And this one, in my mind, is the one that is more relatable to a matrix tablet, because a matrix tablet is a dry oral solid dosage form with dry HPMC particles distributed throughout. Here you have to read the x-axis from the right-hand side to the left-hand side, just like what I showed in that previous slide. You start on the right-hand side with a hot dispersion, where the HPMC particles are dispersed in hot water, for example, and then in controlled fashion in a rheometer, you start to drop that temperature. Here, we look at various 4,000 millipascal second molecular weight grades, and one other one with the A chemistry, right? One additional, but E4M, F4M, K4M. Here, as you work from right to left along the x-axis, you see at 50 degrees Celsius or greater, K4M starts to go into solution. You see a build in the rheometer and torque. Next in line comes E4M in mid to high 40s centigrade. Next comes F4M in low to mid 40s centigrade. And then finally come your A's, your A chemistries. A4M are around 30 degrees centigrade, and then another type of A chemistry at 20 degrees centigrade. The thing that I have to keep in my mind, and I would like you to keep in your minds, is 37 degrees Celsius. Which chemistry is going to give me the best chance of formulating a matrix tablet that'll create that hydrogel readily, quickly around that matrix tablet at body temperature, right? So I want to be at or above 37 degrees Celsius for my powder dissolution temperature. F chemistry, that should do it, right? It starts to hydrate. Its powder dissolution temperature is low to mid 40s. E4M, that should do it even better, right? Mid to high 40s is its powder dissolution temperature. K chemistry, yes, that will do it, right? There's a nice delta between body temp and 50 degrees Celsius or greater, where K chemistry will readily form that hydrogel around that matrix tablet at body temperature. And that, in my mind, is why K chemistry is really the go-to polymer for hydrophilic matrix tablets. So at the base of the pyramid, I really think, I think about it in terms of that. What chemistry is going to give me most ready hydrogel formation at body temperature, where that matrix tablet will form that hydrogel that envelops it. Now, if I look at some controlled release profiles of matrix tablets having the different, 4,000 millipascal second molecular weight grades like A4M, E and F4M, K4M, now it starts to make sense, right? So an A4M matrix in this formulation, this API, the drug released quite rapidly because that A4M did not readily form that hydrogel at body temp around the matrix tablet. So you get more burst release. E4M, F4M, those came next. A little bit of burst release, but they do eventually form that hydrogel around the matrix tablet. K4M, minimal burst release of the chemistries compared in this formulation. Minimal burst release, the greatest extent of controlled release, because that matrix tablet containing the K4M formed that hydrogel so readily that it enveloped the matrix tablet. That's critical, right? For that formation of that hydrogel is critical for robust controlled release performance. So I set that at the base of my pyramid. Now I move to the next layer up, achieving that percolating network of HPMC particles throughout the matrix tablet. I'm not a great artist, but these little circles are supposed to be HPMC particles, right? You want them to be uniformly distributed throughout the matrix tablet, and you want them to be in close enough proximity to each other so that when that tablet is swallowed, it hits the aqueous gastrointestinal media. Those particles at the outermost surface will start to hydrate, they will start to swell, and then they'll start to coalesce to form that hydrogel that envelops the matrix tablet. This can be achieved, or this can be impacted by two key aspects, this percolating network. One is the concentration of the HPMC in the formulation. And here you see a movie that's sped up over 24 hours, where you've looked at 5 formulations of matrix tablet with varying amounts or concentrations of HPMC. 10% on the left-hand side, all the way up to 50% on the right-hand side. And you can see the impact of concentration, really getting a close enough proximity of those HPMC particles next to each other in the matrix tablet on the swelling and erosion of the matrix tablet over the 24-hour time period. As a general rule of thumb, we recommend 30% to kind of achieve that percolating network. It gives you the best chance, as a general rule of thumb, of achieving that percolating network of HPMC particles throughout the matrix tablet. So 30% is typically what we recommend. But you also have to think about the other impactor, and that is particle size. All right, so here, picture a tablet that you're looking at, that you're looking down in a petri dish that has aqueous dissolution media in it, and you're looking at this edge of the matrix tablet, and you're looking at hydrogel formation in the radial direction. You're looking at two different particle sizes of HPMC in the matrix tablet, a fine particle size on the left, a coarse particle size on the right, and then you're looking at two concentrations. So with a fine particle size of HPMC at 30%, no problem, right? You have good, consistent, robust hydrogel formation over this 4-hour time period. These movies are sped up over 4 hours in this slide. 40%, even better, right? But 30% does the job, no problem. With a coarse HPMC, what you can see is that, you may need a little bit more than 30% to achieve that percolating network, because you have quite a bit of hydrogel erosion here, that's just sloughing off over the 4 hours. I'm not saying you have to use up to 40%. Don't get me wrong. Maybe you could use 32 or 35, percent with a coarser HPMC. But you have to think about both aspects when you're thinking about achieving that percolating network throughout the matrix tablet. Well, let's say we have achieved our percolating network. Let's move to the next layer. Now, this is getting to that point where Adriana was talking about. Let's look at the API properties, soluble versus insoluble APIs. Let's look at the release duration, 8 hours, 12 hours, 24 hours. Now we can start selecting our molecular weight grade, right? And here I show 4 of our molecular weight grades of K chemistry, K100M on the left-hand side, down to K100LV on the right-hand side. And again, this is a movie that's sped up over 24 hours, but you can see the impact of the molecular weight grade on how that matrix tablet swells and how extensively it erodes over the 24 hours. And as you might expect, shorter polymer chains like K100LV, those will erode faster. And on the flip side, longer polymer chains like those in K100M will erode more slowly. I like to refer to this as my coarse tuning knob, right? When I'm tuning the duration of the controlled release profile. Or I'm thinking about, do I need more erosion-dominated release with like a more poorly soluble API? Or do I need more diffusion-dominated release with a more soluble API? That'll help me to choose which molecular weight grade. I would use like a K100M for something more soluble, a K100LV for something that's poorly soluble. Now, if you have to, you can keep going right up the pyramid. All right, so we just talked about the coarse tuning knob with these different molecular weight grades. What if we were able to, within a particular molecular weight grade, if you look at this box here for K chemistry in this lower left-hand grid, what if we were able to look at a low HP, mid HP, or high HP K4M? Now, if you look at this movie that's sped up over 24 hours, you have another kind of, quote, knob that you can turn to tune swelling and erosion over the 24 hours. And that I like to refer to as the fine tuning knob, like using the analogy of a microscope. Well, how is that pertinent to controlled release performance? Some of my predecessors, some of my mentors, way back when, they looked at 3 different model systems: paracetamol, diclofenac sodium, salicylic acid. They looked at, they formulated matrix tablets with at least 30% polymer, all the things that I talked about already in the matrix tablet. They looked at controlled release on this y-axis versus HP substitution and substitution. And in this case, they used K15M. And here you see distinctively 3 different response surfaces, controlled release response surfaces, as a function of within chemistry substitution. So it's an API to API case-by-case basis, right? That you might want to think about as you're thinking about that pyramid of robustness. Well, we took the paracetamol example and took it one step further. We had 20 batches of methocell K4M, and these 20 batches of methocell K4M, by the way, also met CR type specifications. We formulated what we believe to be a robust, matrix tablet formulation with paracetamol, 30% polymer, all the things I talked about. We looked at the controlled release profiles of those 20 different batches of tablets containing these 20 K4M batches, and we saw nice, robust, consistent controlled release performance. Towards the end, it looked like maybe the release profiles were fanning out a little bit and getting a little bit more variable. Towards the end of the 24-hour release profile, we believe this to be true because we did go to great lengths to minimize the measurement variability, right, of the dissolution methodology. And so we said, well, why don't we try to model that? Very simple linear model. Let's try to tie performance out at these later time points to some of these critical to functionality attributes of the HPMC polymer. And we were successful. We found, using very simple linear modeling, that HP substitution did, for this formulation, for this API with these 20 batches, did become a statistically significant factor on controlled release functionality, followed by 2% viscosity, which was a borderline significance within the confines of this study. Particle size, since everything met CR, specifications, we achieved our percolating network. Particle size in this case was not a statistically significant factor, and nor was methyl substitution, because methyl substitution just simply did not vary enough. But what we found within the confines of this study, we looped in, obviously drug release performance at the latter part of the profile. We looped back in that powder dissolution temperature rheology technique that I talked about earlier. And we found that as we increased HP substitution along the x-axis, that drug release increased. And you can see, even though it's over a narrow range, it is a clear trend, as did powder dissolution temperature. It increased as we increased HP substitution. So the polymer became more hydrophilic, right, as we increased HP. We took this one step further as I'm getting closer here to the end of my part of the presentation, and we looked at a different API. In this case, we were looking at glycoside, which is a poorly soluble type API. It needs more erosion type release. Here's another case that Adriana talked about, zero order release, with these black profiles are the benchmark target profiles. Zero order release, and we were trying to target zero order release over about a 6 to 8 hour time frame. So that's not something that's typical for a hydrophilic matrix, as Adriana talked about. We typically see more first order release, like with these pink profiles or these purple profiles. But what we looked at is we looked at low, medium, and high HP. So low HP would be purple, medium HP would be pink, high HP would be blue. We looked at low, medium, and high HP materials to see, all right, so which is going to give me the best chance of hitting that zero order kinetics? And it was the blue materials, the high HP materials. So that helped us, phase one, zero in on, all right, we need to be looking at high HP. Okay, this is glycoside. This is more poorly soluble. It needs more erosion-dominated release. Let's look at K100LV and let's look at K4M. So we looked at high HP K100LV. That gave us just a little bit too fast of release compared to the benchmark profiles over on this right-hand side. And high HP K4M gave us a little bit too slow of release. So then at that point, we just did a handful of experiments combining different ratios of high HP K100LV to high HP K4M, different ratios of those in the same matrix tablet formulation. And very quickly, we were able to triangulate using just this template formulation, really just understanding the polymer fundamentals, right? And tweaking the polymer, plugging and playing the polymer to where we were able to zero in on the right kinetics, zero order release over the 6 to 8 hour time frame. And just focusing on the polymer alone, we hit an F2 similarity factor of up to 84. Now, a formulator may take this further, right, and actually change the formulation recipe a little bit and really perfect that, to match it perfectly. But our scope as polymer scientists was really to understand the fundamentals and the criticality of the polymer attributes on the controlled release functionality. So again, I'll try to wrap it all up here so we have about 10 minutes for Q&A. What Adriana started with was she really kind of laid the foundation. She talked about the diverse portfolio of derivatives or of polymeric excipients that are out there for achieving controlled release. She talked about and narrowed in on hydrophilic matrix tablets, why they are so critically utilized, the fact that they are so heavily utilized throughout the industry. They're relatively easy to manufacture. They're universally well accepted across kind of geographies and patient populations. So we narrowed in on hydrophilic matrix tablets for the scope of this presentation. And then on the second half of the presentation, I really tried to give you, within a 30-minute time frame, kind of the pyramid that I have in my mind, tying the structure, property, performance fundamentals, right? Structure, property fundamentals of the polymeric excipient to controlled release functionality. And again, I think about this pyramid. I start at the base, I establish a good foundation, and then I build on it layer by layer. I start by which polymeric excipient will allow, when formulated into a matrix tablet, most ready hydrogel formation at body temp. Once I have that, I move up to that percolating network of HPMC particles throughout the matrix tablet. That gives me my next layer of robustness. And then I can start thinking about the release duration, the API properties. That helps me to select the molecular weight grade. And then finally, I may stop there, but if I need to, I can keep going and I can fine tune the release using things like HP substitution. So with that, I leave you here with our contact information, and I leave us with about 10 minutes for Q&A. I thank you for participating. Thanks, Adriana, for setting a good foundation for me. And with this, I'll stop sharing so that we can go into Q&A. Thank you so much.
Thank you, Drew.
Perfect. Thank you both so much for that great overview. Before we get started on that question and answer session, I would like to quickly remind our audience how to submit questions. Any questions you have can be submitted by typing them into the Q&A box, which can be found directly below the video player. So our first question today is, can you please explain how the burst effect can be reduced when using HPMC as a controlled release agent?
All right, perfect. Thanks. Yeah, maybe I'll take that one because I spoke to that a little bit during the presentation. It's really about that pyramid that I was talking about. You saw the example that I showed with the different chemistries, A4M, E4M, F4M, K4M. Start by thinking about why does the burst formation occur? It occurs because some of that drug escapes from the matrix tablet and is released prior to the formation of that hydrogel that envelops the matrix tablet. So as I step myself through that pyramid, I want to think about, all right, which polymer most readily will form that hydrogel, around the matrix tablet. That slows down that burst right there. But I also have to think about those other aspects of, do I have a sufficiently percolating network of those HPMC particles throughout the matrix tablet? And then I just keep working my way up that pyramid. But this is a very relevant question because that formation, that ready formation of the hydrogel around the matrix tablet is so critical to minimizing that burst release, but then thereafter modulating the release of the drug, for the rest of that time interval.
Fantastic. Thank you very much. Moving on to our next audience question. In an HPMC-based extended release formulation, HPMC K100M lower strength F2 is not matching with higher strength, even though both are dose proportionate. I don't know if someone could weigh in on that.
Yeah, I can. Well, actually, there were a word that I mentioned and I really highlighted was it's a surface. How can I say? It depended on the surface against the volume we have, right? Then if you have exactly the same formula in different relations, surface volume, your surface is changing. Then probably the smaller one have less surface than the big ones and will be closer in the profile. And that is very typical. And you can work, of course, the ideal thing is to maintain exactly the same formula, because it will be cheaper, all the bio studies, then probably you can work with the tooling supplier because it is very difficult to calculate. If it's just a plain tablet, it could be more easy to calculate. But if it is a complicated shape, probably the tooling supplier can support you in making that calculation and trying to achieve the same ratio between volume and surface exposed.
Fantastic. Thank you very much. On to our next question. What criteria should be considered when selecting excipients for controlled release formulations?
You want that one, Adriana? Or you want me to take it?
Yeah, we can take it.
Okay. So I think about, what criteria when formulating controlled release formulations? I think about things like, well, the API dosage in the formulations. I've seen graphics where, it shows kind of like a suggestion grid. For example, if you're working with a low dose API, a multi-particulate type of a formulation may well be within scope for you, or an osmotic type of a formulation may well be within scope. If you're working with a really high dose API, well, that makes it very difficult to load a multi-particulate sufficiently with the API itself. Or same thing for an osmotic type dosage form. Matrix tablets, they tend to be a little bit more flexible and forgiving, as a function of your API dosage, how much of it is in the formulation. I've seen formulations of matrix tablets that have up to like 75, 80% API in there. It's not the best case scenario, right? But I've seen it. Easily you can go up to 50% in a matrix tablet formulation, whereas maybe you could only go up to 10, 20%, something like that in another type of a controlled release delivery system. So one of the criteria I think that's key is the API properties, the API dosage. Adriana, anything you want to add to that?
It's exactly the same. Yeah, and there is another characteristic going deeper, like a flow, depending what are you planning to do and what are your options in the plant. But yeah, it's a complicated question.
Yeah.
Have a lot of answers.
Fantastic. Thank you both. And then, are there advantages to combining E and K HPMC to modulate release?
I can take that one because I did it. Actually, for me, it's easy to go with just a polymer because you have some variability associated, as True show us. Inside the box of HP, for example, if we go to USP criteria is very broad, then we go with some product going just narrowing it down, and that if you have both two polymers, you have like two variabilities. Then for me, it's better to try to get a formula with just one. But in some cases, you need this fine tuning. If we cannot achieve with this special product, like more narrow it down the HP, for example, particular site, you can have a combine of chemicals. With A, for example, if you use in the formula, the shell will support the shell formation, for example, for the key chemistry, but it would be the release will be more open. You will have more faster actually. Then it's possible to do. My advice would be to try to keep one, but it's a tool that you can use.
Perfect. Thank you. Would you like to add?
No, just to give True. Yeah, if you have some experience with that.
Honestly, I didn't, so I'm glad you answered that one, Adriana.
We're a team.
Thank you. And then, are the cloud points of all grades the same?
I can take that one. No, they aren't. So hopefully I showed in that one slide, that graphic on the left-hand side. The cloud points, in my experience, kind of trend with the precipitation points or the gelation points. So that is a fingerprint that is unique from chemistry to chemistry. So the cloud points will differ based upon the substitution chemistries, and they can even, fine tune, adjust a little bit within the chemistries as well. So no, short answer is no, they aren't the same for all of these different chemistries of water soluble cellulose ethers.
Perfect, thank you. And it looks like we have time for one more question today. Does wet granulation influence the particle size of HPMC, so starting material?
Ooh, I have a good one on that, but I'll let you start, Adriana, since we're kind of going back.
Yeah, actually, it doesn't. If you make a good granule, when you compress it, it will recover the initial shape. Probably the hardness could be different, but at the end of the day, you will have the same profile. That is my experience. If you over granulate, probably the tablet will be not good enough, and you have a different profile. But in general, if you make a good granule, you have the same profile. Of course, the particle size would change, but not dramatically for affecting the profile once it's compressed. True. If you want to add something.
You hit it. You don't want to over granulate, right? Because then you could start influencing the percolating network of those HPMC particles, right? The only thing I'll add there is it also matters whether or not you include the HPMC, since we're talking about hydrophilic matrices, you include the HPMC intragranularly, so within the granules during wet granulation, versus extra granularly. So you make your granules with your drug, for example, and then you blend in your HPMC powder afterwards. I've seen both, right? And some formulators even use a combination of some HPMC intragranularly and some extra granularly. So that's the other factor that I will, will add in there for consideration.
Sure. Good point.
Perfect. Thank you. And thank you both so much again for that great presentation. It is time to already wrap up, so I want to start by thanking the audience for attending and participating in today's event. And I'd also like to thank our sponsor, IFF Pharma Solutions, for making today's educational webcast possible. We would also like to ask everyone in the audience to participate in a brief survey that will appear on your screen after today's presentation has ended, and you will also receive an email alerting you when this webcast will be available for replay. And we invite you to forward that announcement to your colleagues who may have missed today's live event. We hope to see everyone next time. Thank you and take care.